Imagine never having to remember to take a pill or endure a painful injection again. Scientists have just developed a tiny living pharmacy implant that sits under your skin and manufactures your medications 24 hours a day, 7 days a week, and it is already working in the lab. Today, we will examine a major leap in biotechnology known as the Hybrid Oxygenation Bioelectronics System for Implanted Therapy, or HOBIT, co-led by researchers at Northwestern University, Rice University, and Carnegie Mellon University. This device acts as a self-sustaining pharmacy inside the body. It is roughly the size of a folded stick of gum. The research funded by DARPA and Breakthrough T1D was published in the journal Device on March 27, 2026. To understand its significance, consider ordinary pills or injections. Traditional methods deliver discrete external doses of medicine, which inevitably cause peaks and troughs of drug concentration in your bloodstream. This often leads to high side effect risks during peaks and ineffective periods during troughs. The HOBIT implant works differently. It uses genetically engineered cells to continuously produce biologic drugs at a steady rate directly inside your body. This represents a massive breakthrough because it solves the historical oxygen bottleneck. Previously, when scientists tried to implant high-density therapeutic cells, those cells would quickly starve of oxygen and die in the subcutaneous tissue. This is the first successful demonstration of a fully integrated wireless biohybrid platform capable of keeping these cells alive to sustain multiple biologic drugs with different half-lives for weeks in animal models. To grasp exactly how this tiny device achieves such a complex task, we must look at the specific engineering that keeps these therapeutic cells alive and functioning. How do you keep millions of engineered cells alive inside the body without them suffocating? The answer is the HOBIT, a bioelectronic synthetic biology hybrid. According to a recent MIT study, this device combines genetically reprogrammed living cells with active electronics, including wireless communication chips and an onboard battery. Inside the implant are designer cells, mammalian cells densely packed at approximately 60 million cells per milliliter within alginate hydrogel beads. A semi-permeable membrane shields these cells from the immune system while permitting nutrient inflow. However, subcutaneous tissue lacks sufficient oxygen for this high cell density. To solve this, the HOBIT features an onboard oxygen generator called HOO2. Using iridium oxide electrodes, it performs electrolysis on surrounding tissue fluid, splitting water into hydrogen and oxygen gas via the equation 2H2O yields 2H2 plus O2. This continuous delivery of pure oxygen keeps the cells metabolically active. The study reports that oxygenated implants maintained approximately 65% cell viability over 30 days compared to just 20% in non-oxygenated controls. Consequently, these cells continuously transcribe genetic instructions and secrete targeted medicine 24 hours a day, entirely eliminating the need for repeated patient dosing. Keeping the cells alive is only the first part of the equation, as the true power of this implant lies in its ability to manufacture completely different classes of medicine simultaneously. Treating complex chronic conditions often requires polypharmacy, which is the use of multiple drugs at the same time. The challenge with biologic drugs is that they have vastly different half-lives. A large antibody might linger in the body for weeks, while a small peptide might clear the bloodstream in hours, making it nearly impossible to maintain stable levels using external injections. Researchers bypassed this issue using synthetic biology. They engineered a single population of cells with genetic circuits utilizing polycistronic expression, allowing these cells to produce multiple distinct therapeutic proteins simultaneously in balanced ratios. In their trial, the implant produced 3 specific biologics at once. These included an anti-HIV antibody, a GLP-1-like peptide analogous to enotide for blood sugar and appetite control, and leptin, a metabolism-regulating hormone. These specific drugs were chosen as a rigorous stress test for the platform because they have vastly different sizes and mismatched half-lives. Remarkably, the implant kept all 3 drugs at stable therapeutic levels in rat bloodstreams for a full 30 days. This multi-drug output makes the platform incredibly versatile. Because the genetic circuits can be swapped out without redesigning the hardware, a single device could be programmed to treat complex, overlapping comorbidities automatically. With the ability to synthesize multiple complex therapies at once, this technology is uniquely positioned to address some of the most difficult and prevalent medical conditions. Why do breakthrough treatments often fail to improve patient health? The answer is frequently the delivery method, not the drug itself. The prime candidates for this living pharmacy technology are chronic diseases requiring long-term biologic therapies where poor patient adherence undermines efficacy. Consider type 2 diabetes and obesity. Currently, patients rely on frequent injections of GLP-1 agonists. This strict regimen induces injection fatigue, leading to missed doses. The HOBIT implant shifts this paradigm. By utilizing engineered cells to continuously secrete therapeutic proteins, it eliminates the need for daily or weekly shots. Capitalizing on this mechanism, the ARPA-H-funded ROGA project is actively developing a pill-sized version of the implant specifically for metabolic conditions. The potential extends well beyond diabetes. For autoimmune disorders like rheumatoid arthritis, the implant can continuously synthesize and deliver complex monoclonal antibodies. For type 1 diabetes, HOBIT provides the precise vascular integration necessary to sustain high oxygen demand pancreatic islet cells. This continuous delivery solves a systemic healthcare failure. According to the World Health Organization, roughly 50% of chronic disease patients miss their scheduled doses. Medical literature demonstrates this compliance failure contributes to an estimated 125,000 preventable deaths in the United States annually. By deploying a set-it-and-forget-it implant, healthcare providers can remove the logistical burden of remembering medication, ensuring patients maintain precise therapeutic levels without interruption. While the potential benefits are massive, placing a living drug factory inside the human body introduces unique risks that engineers must carefully mitigate. How do you control living cells once they are inside the human body? While bioelectronic implants promise autonomous treatment, they introduce three severe risks: immune rejection via the foreign body response, cellular death from oxygen starvation, and uncontrolled proliferation. To neutralize these threats, the device utilizes a two-step fail-safe architecture. First, a two-stage encapsulation membrane physically isolates the therapeutic cells. This barrier shields them from the host immune system, preventing rejection without requiring systemic immunosuppressive drugs. Second, the ECO2 generator prevents hypoxia by maintaining a constant, localized oxygen supply. What happens if these cells produce too much medication? Researchers use oxygen as a biological throttle. By wirelessly adjusting the electrical current to the ECO2 generator, doctors can modulate the oxygen levels inside the chamber. Because synthesizing drug proteins requires high metabolic energy, lowering the oxygen supply directly reduces the cell's production capacity, safely dropping the drug output. If the cells fail entirely, medication delivery simply halts. In the event of a critical malfunction, the device requires straightforward extraction. Because it is implanted subcutaneously just below the skin, doctors can retrieve it via a minor outpatient procedure. A recent safety study demonstrated this protocol. Researchers implanted a cell-free version of the device into a cynomolgus macaque and safely removed it after 1 month with no major complications. For an absolute biological fail-safe, earlier DARPA-funded research developed genetic kill switches. In a systemic emergency, a remote signal could trigger these engineered genes to destroy the cells, instantly shutting down the living factory. Despite these sophisticated safeguards, the journey from a successful laboratory prototype to an approved medical treatment is a long and rigorous process. How close is the HOBIT platform to actual human patients? While the results appear revolutionary, this remains strictly a preclinical animal model breakthrough. According to the foundational study, the device demonstrated 30-day efficacy solely in rats. No human trials have been initiated, although the research team is consulting the FDA regarding a structurally similar localized ovarian cancer implant. The HOBIT multi-drug platform remains far from real-world clinical use. Transitioning to human therapy presents three concrete barriers. First, engineers must prove device durability over several years rather than weeks. Second, they must evade chronic foreign body immune responses, specifically the tendency of the human immune system to encapsulate foreign objects in dense fibrotic scar tissue. Third, the transition requires establishing good manufacturing practice or GMP facilities. Producing clinical-scale biohybrid devices involves massive logistical complexity and sterile manufacturing lines. Because of these hurdles, human applications are realistically 5 to 10 years away, strictly pending successful large animal trials and meticulously phased human safety studies. Even with years of testing ahead, the foundational success of this implant signals an inevitable and permanent shift in how we approach healthcare. What if treating a chronic disease meant installing a microscopic pharmacy directly into your tissue rather than relying on daily injections? This contrast defines the ultimate paradigm shift in modern therapy. transitioning from taking medicine externally to making medicine internally. According to clinical data published in Nature Biomedical Engineering, traditional biologic therapies suffer from a 50% non-adherence rate over 2 years due to injection fatigue and cold chain storage failures. The implantable biofactory solves this by utilizing the interstitial fluid of the patient. First, the semi-permeable device draws in native water, glucose, and amino acids from the surrounding tissue. Second, engineered cells within the implant use these raw nutrients to synthesize specific therapeutic proteins. Finally, a programmable microfluidic valve releases exact dosages into the bloodstream on a precisely tuned schedule. By acting as a personalized, programmable drug factory, this technology bypasses the global supply chain entirely. It has the potential to replace daily pills and weekly injections for conditions requiring monoclonal antibodies. Ultimately, this transforms long-term disease management from a reactive daily chore into a proactive, embedded biological process, significantly reducing systemic healthcare costs and freeing patients from the interruption of complex medical regimens. This fundamental change in drug delivery will redefine what it means to live with a chronic illness. The Northwestern HOBIT implant proves that living pharmacies are no longer science fiction. By successfully combining synthetic biology with wireless bioelectronics, researchers have created a blueprint for the future of medicine. While human trials are still years away, this breakthrough promises a massive shift in healthcare. We are approaching a future where managing complex chronic diseases like diabetes or HIV is as simple as a single set-it-and-forget-it implant that turns your own body into a drug manufacturing facility. Thank you for watching, and stay tuned as we continue to track the medical breakthroughs shaping our future.